we will have the privilege of having Mike Lewicki presenting about neural stuff. So Mike is a professor at the EECS at Case. He received his bachelor from Carnegie Mellon University, PhD from Caltech, did some other stuff, did the tenure at Carnegie Mellon before moving here, and his research work focuses on understanding neural information processing and computational principles in sensory coding, perception, and scene analysis. So looking forward to hearing nice stuff from Dr. Lewicki, and please welcome.
Is this thing on? Okay. All right, so thank you to Hadrian, and actually the whole HPC. A lot of this research wasn't possible without the HPC and their staff, and they've been extremely helpful. So my whole career, really since I first visited a university, I think I've been fascinated by the brain because I walked into a university bookstore, and it was the first time that I had seen all these academic textbooks, and I found this one called Thinking, Problem Solving, and Cognition. And it was my introduction to like the brain, and at that time I was kind of set on artificial intelligence. But my career path is kind of wound around in that space, and I've always been fascinated by the brain, and so I keep coming at it from different approaches. And so this talk ties in because so Mark talked about like the HoloLens, right, and our interpretation of 3D structure. So this is a brain. We're looking at a 2D image of a simulated 3D image of a brain looking at a 2D image. There's a lot of recursion going on there. And the question we have is, well, how does the brain make sense of the world? Because you can trace the, can I do this? Oh, this will work better. So you can trace the information, right? And anatomists have mapped out neural areas, and Mark showed beautiful pictures of all the connections. But what are those connections doing? What are the computations that make it possible for us to perceive the 3D world and reconstruct the 3D world and understand it? So that's what I'm going to try to give you a sense of in this talk. And the study of brain function involves a lot of different areas. From the high-level stuff of cognitive science and perceptual science down into the neuroscience of what the neurons are doing. And then there's the theoretical side of it, like the mathematical, where it's artificial intelligence, machine learning, computer vision, and so on. And my own research has kind of wandered around in that space. And what I try to do is take a theoretical approach to understand what are the principles of neural information processing. So could you understand the fundamental computations that all perceptual systems would have to do in order to solve certain problems? And I came to this way. So, I started off kind of at a high level, artificial intelligence, robotics, and then I worked my way really down low. And so this was during my PhD when I was studying neural representation in songbirds. How do they represent song? And this is an actual neuron that I stained myself, and it represents, it responds selectively to the song. And if you play it songs, which is what's shown at the bottom here, it responds in a very specific way. And this was amazing, because look how detailed that response is. But after this kind of novelty wears off, you're left with the question, well, what does it do? Right? How do you figure out what it does? And when you're a neuroscientist and doing physiology, you sort of spend all your time thinking about individual neurons, and you're trying to ascribe meaning to them. But you come up against the limitation that, well, you don't really know what a neuron is doing in the context of the whole system. And how would you go about figuring out what the system is doing anyway? So we have to pull back and start asking different kinds of questions. And this field, in my own research, is motivated by a very prominent figure who started the field of computer vision, which made the HoloLens and things like that possible. And he was also a computational neuroscientist. And what he said was that an algorithm is more likely to be understood by understanding the nature of the problem being solved than by examining the properties of the system. Right? The properties will only get you so far. And at some point, you have to understand, well, what is it trying to do, and what's the optimal way to solve that problem? And you might think, well, in the brain, this is kind of a hopeless task, right? But there's hope, right? Because we have physics, right? And physics was faced with the same problem. When you look out at the world seems hopelessly complicated, but yet it obeys rules, right? There are laws of gravity, for example. And if you were to look at eyes, so there's a million different eyes in the animal kingdom. And you might think, how could they have anything in common, right? And if you didn't know, say, about optics, you would have no idea. They're just, you would have no idea what they're even doing. And if you go back in history and look at what people thought about visual perception before they understood optics, it was crazy. They thought that the eye projected things into the world, and that these beams somehow connected with objects, and that's how you recognized an object or a person. But now we understand light, and we understand refraction and things like that. And so when you look at this, you say, I know what's going on, because you understand optics, right? There are fundamental principles, and you can say that, well, there's a pinhole type eye. There are eyes that are refracted. There are reflective eyes. There are eyes that work on shadows. But they're all based on the same principles, right? So these principles are what allow you to understand the complexity, because fundamentally, similar things are going on. There's a lot of variation, but they all obey the same set of rules. And so when we look at something like the brain, we want to ask, well, what are the rules? What are those principles? And if you understand this, you can kind of understand how evolution might work in this kind of space. Here's a simulation of the evolution of an eye, right? And it's starting from just an array of light-sensitive cells. And they simulate just a few % change generation to generation. And it doesn't take too long, in this case, 364,000 generations, but it evolves first a pinhole eye, and then it starts, you can also change the density of this refractive material a little bit each generation, and eventually it evolves a lens. And each little step is an improvement in fitness. And the system doesn't know, quote unquote, anything about optics, but yet the system is constrained to the laws of optics, right? And that's what forces the system to take the shape it does, to have the properties it does, because there are these physical things that it's obeying. But if we think about perception, right? Well, optics is one part of it, right? That's the camera lens. But then what happens after that? Once the information falls on the retina and begins to be processed, what goes on? Well, that's where computation comes in. And this is a project that I did with a postdoc here at Case, and this was done on the HPC. And that last example, right? Part of what you're doing when you're trying to understand the neural code is do simulations and try to find the optimal solution. So in a way, you're recreating evolution. But when you do that, you also have to recreate the environment, because you're trying to figure out how is the system optimally adapted to the environment. So before long, you realize, well, we really have to simulate the whole universe. So there's no end of computational need. But we have to simplify it, right? So when we look at something as complex as the retina, we have to ask, what are the key features that allow the neurons to form a code? Because the light goes in, the optic sensors are at the back, and then the circuitry to send the signal to the brain is in the front, and that goes down the optic nerve, and then that's how your brain sees the world, right? Based on those signals. But they're not just sending the raw, photoreceptor signals. They're computing. They're computing things, and they're encoding it in a way that we want to understand. And so when we look at it, well, we know that neurons can represent things, right? They're sensitive to light, for example. We also know from anatomy that they change in density. For example, at the fovea, you have a lot of photoreceptors and relatively little amount of circuitry, whereas in the periphery, it's the opposite. The photoreceptors are much more spread out, and there's a much greater convergence in terms of the circuitry. So that's something we can incorporate into our model. And there are also a number of limited neural resources, and that turns out to be a very important part when you're trying to answer the question, what do neural circuits compute? Well, the space of possible computations is huge. So you have to understand what the constraints are, and one of the biggest ones is energy, right? It's expensive to compute, and the brain and all neural systems are very constrained in how much energy they have available to do computation. And that's similar to the constraints of how much information each neuron can represent. So when we represent a neuron, we don't represent it as a floating point number, right? You can't represent that. You can only represent about 1 bit of information. So the way neural circuits compute is very different from the way we might compute in, say, a silicon chip, right? The principles are different. So we take this system, and now we have to build a model, all right? So we model the neurons with neurons. So this is a neural network. And then we have the original signal. So this is just a 2D vector representation of it, or sorry, 1D. And it goes in, it gets projected onto the back of the retina. And then there's noise and blur as part of the optics. And so we model that. And then these are that's the back of the retina. So this is where the phototransduction happens. And then there's neural noise, right? That represents the limited precision. So when it responds to a signal, it can only encode so much information about, say, the intensity of light falling on that particular location. And this last step, the decoding step, is not really part of the real system, but it's how we train and optimize the model, right? We want to optimize it so that we maximize the information transfer down the optic nerve. And so if we've maximized it, we should be able to take that signal and decode it so that we reconstruct as much of the original image as possible. Okay, and so adapting the model means we take this part here, and those are what we're modeling by these weights. You know, we're taking, basically adding together the responses from different neurons, you know, multiplying it, things like that, and we're computing something from that. But when we optimize it, we're going to search through this whole space of possible computations using optimization and derive what's optimal for our goals, which is to, given the system, minimize the error or maximize the amount that you reconstruct the signal and minimize the reconstruction error. So this is the model part, all right? That's the neural encoding. So what we do is take natural images, we take this model, and then we adapt it. And this is the thing we ran on the computers over and over, because we're really simulating evolution, right? We simulated the evolution of the optics. Here we're simulating the evolution of the retinal circuitry. And we're saying, what's the optimal retinal circuitry? And if we understand the principles, the information processing principles, there should be convergence. We should be able to predict what the retina is actually doing because we understand its function. And that turns out to be true. So here now is a two-dimensional picture of a receptive field, and these represent the weights of a single ganglion cell. That's the cell that sends the signal down the optic nerve, and how it's connected to all the different photoreceptors, right? So each cell has possibly hundreds of connections, and those weights are shown here. So those show positive influencing weights, and these are negative influencing weights. And the gray ones are 0. That means there's not really any connection. And so in the fovea, the weights are very tight. They represent higher resolution information. But they don't just encode the photoreceptor. They encode the photoreceptor's signal here and then the difference in the surround. So that's its way of encoding the information. But if you go out to the periphery, the side of the vision, things are much different, right? It's a much broader integration of the photoreceptors. And down here you can see how things are organized in a population. So at the fovea, it's very tight. Right? So it's very much like a pixel array. And you can see the receptive fields are small. They're laid out in a grid. That happens naturally. And in a periphery, since you have fewer neurons, it has to spread themselves out. So they kind of take on this arrangement. And that's what you see in the actual retina. And what's really interesting is that this organization is what physiologists have measured and neural anatomists have measured in the actual retina. But they've also done the physiology and how this code changes to different conditions. And we can also simulate that. So, for example, the code changes depending whether the light is bright or dim. Right? So you all probably have the sense that when the lights are dim, you can't see as well. And that's because the signal to noise is much lower. And if we change the light now, the optimal code changes. And so if it's medium light, it starts to spread out. The periphery code doesn't change. As it gets to low light, we sort of lose the inhibitory part. It doesn't do as much subtraction. If we have very dim light, it basically just does an average, right, to reduce the noise. And that behavior is exactly what you see in the retina. But here we've derived it theoretically. So what we can say is that, well, the optimal computation, if you want to minimize reconstruction error to send the maximum amount of information given the system, is to have this type of code under bright conditions, and then that type of code under dim conditions, and then gradually change between them. Right? So it reproduces that part of the physiology. You can also contrast this with a code that an engineer might give you. So I have to be careful. I'm in an engineering department. And people have studied coding for a long time. Like whenever you take a picture, you have a JPEG, right? That's a code. So you have a discrete cosine transform. You could do a wavelet transform. And those are, in some senses, considered optimal codes. But they're not optimal when you have noise in the system. And so the situation is different. And if you take one of those optimal codes and restrict yourself to 1 bit of precision for each coding element, you get terrible reconstruction. And you might think, well, maybe you can't do any better, right? Because you don't have much resolution. But we can contrast this now with our optimized code, where it's the same number of weights, the same space of circuitry, and ask how much better can you do if you optimize it given the constraints. And it turns out you can do a lot better, from 30% error down to 3.8. And that's just doing the right optimization function. Okay, so we have kind of a theoretical methodology now. We have the natural environment, and we have what evolution has done, optimizing its system to maximize fitness. And the systems that are operating in that mode take on certain properties. And what we want to do with theory is recreate that, right? So we want to understand what the optimal model is, what the system is doing. And when we do that, we make a prediction. We don't know what's going to happen, right? We're not modeling the data in the sense that, we're fitting like a regression or something like that. We're really making a prediction. We're saying, what's the best answer? And it might not be correct because your model is wrong. And this actually, I didn't tell you the whole history, but it's not obvious how to get the right prediction because there are so many different things you could predict. You have to have everything kind of in the right regime. So you don't fit to the data. You only compare after you optimize. So it's a functional account or a theoretical account of the data. So this is great, right? And one question you have is, well, how general is this? And this is something that I actually did earlier, but this same kind of theory also applies to the auditory system. So the auditory system has a similar kind of problem. Your tympanic membrane senses vibration. It gets transduced by the middle ear. And then you have this elaborate system, the cochlea, which is responsible for the transduction of sound. And like the optic nerve, your whole perception of the world depends on the code coming down this auditory nerve. That's the brain's connection to the world. So the question you have is, well, what's that code, right? That's how you hear. And so what is the cochlea doing? And so there are different approaches to this, right? People have made their career modeling the fluid dynamics of the cochlea and the basilar membrane and the transduction, all of that. It's very complicated. Can we take a functional approach where we state the problem from an information processing point of view and say, well, what should the cochlea do if it were behaving according to an optimal information processor? And so I'll kind of skip over the details, but what we did is we went out in the world and recorded natural sounds. And we set it up the same way, right? We have this model, we encode things, and we have to be a little bit careful of the kind of code we make. And so the code we make involves action potentials and coding of neural events, or acoustic events, which is very different from how you would do it in engineering, where engineering you kind of have like a linear transformer, you block up the data and transform each chunk. But here we're doing a continuous encoding, but encoding discrete acoustic events. And what turns out to be important is the shape of the acoustic encoding elements, which you could think of like as acoustic features. And just like before, we start off with no preconceptions of what this code should be. These shapes could be anything they want. The answer is whatever best describes the data. And now we do this evolution simulation. All right? And so what's going on here is that the entropy is decreasing, meaning the code is getting more efficient. But the signal-to-noise ratio, that reconstruction error, is getting bigger. So you're using less bits to encode more information. And what happens here is that you get a code that looks very much what people have measured in the auditory system. Let me just back up and show the simulation again. Right? It converges to something. And you can start it from many different locations, give it different kinds of data, but it converges to something very similar each time. So it's like that's the solution that nature has arrived at. And so let's compare it to nature. And so physiologists have measured these response properties of the auditory nerve. You can think of these as like filters. They're time domain filters. And so they have a rapid rise and slow decay. And what they encode best is like a little ping. It almost sounds like a raindrop when you play it. And so these are what we derived. But now let's compare it to a cat auditory nerve database. And if we say, well, what matches closest, what we find is that for every cat auditory nerve filter, we can match one to the theory. And what's amazing about this is that each one of these points here is a free parameter in the model. You're really deriving the whole thing. And yet, it recreates not only the individual time profile, but also their organization in the population. And it depends on the data. And so there's another interesting story how not only is this optimally adapted to our natural acoustic environment, it turns out that if you play this to speech and ask what's the optimal code for speech, you get the same answer. And so that gives you the insight that, well, maybe speech is adapted to our natural acoustic environment and not the other way around. Like speech is fundamentally mimicking the properties of our acoustic world, what we need to do to localize sounds and capture prey and avoid predators and things like that. So the theory works again. And again, we have this general methodology where we define the problem, we specify a model and algorithm, and then we optimize it to natural signals. And we can analyze the results just by comparing the two experimental data, but also measuring the performance, the coding efficiency, other things. And so I'll kind of briefly go through a couple other things that we've done to kind of give you a sense of our current direction. So this gets you into coding, right? But what happens after coding? How do you actually start doing perception? And so we want to go beyond linear features. And here's where it starts to get much more complicated to define the problem you're trying to solve. But one way that we can do it is to talk about, well, you need to form more abstract quantities, right? So when we look at the world, we don't just encode it, right? We describe it. We describe the world in terms of abstract features. So we need to get from the level of pixels to the level of concepts. And there's a bunch of intermediate steps in between. So this picture shows you the weights to these simple features. You can think of these like edge detectors in the scene, but then how do you integrate those edge detectors to detect a contour? And so here's an example of how these linear features aren't able to distinguish any of these classes. But we look at this and we see, well, that's a tree boundary, right? Or this is tree bark, or that's foliage. But the linear features can't distinguish any of those categories, right? They're statistically indistinguishable. However, if you do use the abstraction features, they all fall neatly into categories. And this is another story, but it turns out that this also explains a lot of properties in primary visual cortex that people haven't been able to understand. It's a functional explanation of a lot of these nonlinearities. So it's kind of a validation of the theoretical approach. So now I want to go in a little bit into computer vision, because once you start to get into higher levels, you're really asking the question, well, what problems should you solve? And so we have to look at the kinds of things we do and approach them in a way that we have still some connection to the brain. Like, how would the brain be solving, or neural systems be solving this problem? And so one question we got interested in is the idea of scene analysis. When you look at all these scenes, you can say that some things are similar and some things are not, kind of in the sense of that last picture. All the edges or the images along, say, an edge are different, but there's some sense in which they're similar because you're able to recognize that abstract contact. And what we would like to do is kind of organize these in some representational space. And so then the question is, well, how do you learn this representational space? You know, that these are all, say, mountains, and those are beaches, and these are buildings, things like that. Well, the basic problem that you have, and that you really appreciate trying to do computer vision, is that visual similarity, perceptual similarity, is not at all the same thing as feature similarity. Like, you could look at these images and say, well, they're, you know, it's the same scene, right? You can easily recognize that. But how, right? The features here are totally different from the features there. And it's the same thing here. This scene here is just the same thing, but shifted over. But the features are in a completely different location. And the problem we have is, well, how do we take both of these images and encode them in a way that maps them onto the same space? And what we want to do is that we know that if we take standard approaches, and if we just try and describe an image or a scene in terms of features, that things that we call similar to the feature representation are actually very different. Like, they're in different parts of the space. And for example, these, you know, it's a horizon, right? But the horizon is in a different location. So you might have the same kinds of features here, but how is it to know that, well, those features should be called equivalent to those features? Like, that's true for this context, but in some other scene, it might be not true at all. And so the model has to sort all of this out. And the approach that we came up with was to use physics, right? We believe that neural systems are adapted to the physical properties of the world, and one of that is just perspective projection. And if we take perspective projection and normalize it, right, and so we can formulate that as a computational problem, what we can do is undistort it in a way. So we can map you know, a scene like this by inferring what the camera's perspective is and map it onto some kind of canonical underlying image. And if we did that, now we've done this complex computation, this inference process, by mapping what used to be different onto something that's now similar, and we can compare in a much more regular space. But the problem is we don't know what these are to begin with, right? So it's like a bootstrapping problem. How do you know what these canonical images are before you can do this normalization step? Well, that's the thing that machine learning is good at, right? You start with a rough answer and you refine it, and you keep refining it until you have a very detailed answer, and you can do a very detailed process. And so what we did was we took a large database, and this is something we also trained in HBSP, burned a lot of GPU hours on this. And so there's like 100,000 images and many, many categories. And what we were able to learn was a set of canonical images, so that if you took any image, you can undistort the perspective and translation and scale onto one of these canonical images. And these were automatically learned as part of the scene. And to give you a sense of what that is, like, here's a canonical image, and these are all normalized images. It doesn't mean the images are the same, but they're now more similar so that you can compare them without worrying about the features so much. Here's another set where it kind of expects some kind of object in the middle. And so what this does, it will shift objects to the middle. So it's almost like an intentional process or an eye movement. But of course, our system isn't that complex. And you know, now once you've done that, now it's much easier to compare across features. Here's another example. So you can see that the canonical images even captures kind of quasi three-dimensional relationships, like roads and layouts of the scene. So when we have this, what we can do is do object querying, right? We can take a scene and say, well, what are the closest scenes to this scene? And here are some different comparisons, and the most popular one is deep learning these days. And if you give it this scene and ask, well, what are the closest images, we see all of these, and they're not bad. But you always have to interpret this in context. Well, how do other things do? So here's a different model, and well, in this model, a corn field is just as similar to this room as all of these other scenes, right? And like for the deep learning network, you know, men in front of a fire truck is just as similar to that room as say, these guys playing banjos. And it gets weirder. So you've probably experienced this if you've used any kind of image comparison, right? Paper clips can be similar to an office room. But the model doesn't do great. We don't expect it to do as well as we do because it's not as complicated, but it's at least a step in the right direction. All right, so let me close there, and I'll skip over this part of our current work where we're trying to do motion. And I want to close by just acknowledging all the people involved in this. First, the HPC basically took the maintenance of computers and all the technical issues out of the equation and allowed us just to focus on the research, which is great. And I was coming from Carnegie Mellon and being downstairs from the supercomputing center, which I actually never used because it was just too complicated. And here it's just simple, and that was great. And Ezaburo worked on the retinal coding. Vivian Ming, formerly Evan Smith, worked on the auditory coding. Jan Karklin was the higher order features. Wu Young was the scene normalization. And then my current graduate students are carrying on the flame, Jing and Morgan. And then Michael Schaefer and Josh Tang were undergraduates here and did a great job of getting that graphical database together. All right, so thank you.
Is there any question?
I was wondering about the simulation for the retina. Oh,
Thank you. I was wondering about the simulation about the retina. If you let the simulation go on, can you see like a future step to the development of the retina above what we have as human?
Yeah, first you get like a nautilus, and then you get flies, and then comes the octopus, and then us, amazingly enough. No. What happens is that it optimizes to a fixed point, right? And that fixed point is the optimal code, right? So you can start it from many different conditions. And what's interesting is that fixed point changes depending on the different conditions, all right? So if we say give it more bits for the neural representation, you'll get a different code because that's what's optimal for those conditions. It's like when we said 1 bit of information, that's because that's what neurons can encode. Or when we say, well, the retina, in our example, has a fovea, right? It's concentrated, where you have high resolution. Not all animals are like that, right? Some animals have a very even distribution of photoreceptors, and we can put that in too, and it basically optimizes to a different position. So it's like you're able to explore this whole space, but only at the points of optimality.
Thank you.
That was a great talk, and I learned so much about how the brain works, actually. So, because it's time, we need to shift to the next speaker. Thank you so much again.
Thank you.
So the next speaker is
Dr. Phoebe Stewart. She works at the pharmacology department. She graduated from Harvard and then UPenn. She was a faculty at UCLA and then Vanderbilt before moving here, and became the professor here and also the director of Cleveland Center for Membrane and Structural Biology. So we are excited to learn today about an innovative imaging approach for naturally occurring and engineered nanoparticles. And with that, let's welcome
Dr. Stewart.
Thanks, Adrian. Well, I'd like to thank the organizers for the opportunity to present and also echo what the other speakers have said. We really, really appreciate Roger and his whole team for the HPC Center here. I think what we do just wouldn't be possible if we didn't have your support. And thank you for responding to all of our million requests all of the time. Thank you very much. So now what I'd like to talk to you about today is what we're doing in Cryo-EM imaging, which is a novel way for looking at biological assemblies. These can be viruses, like naturally occurring nanoparticles, or they can be engineered nanoparticles. So I'll start with a brief introduction. What is Cryo-EM, or cryo-electron microscopy? And then I'll just give you 3 short research vignettes to give you an idea of what we're doing in the lab. So this is a YouTube video I found online by one of my colleagues at Scripps that gives you an overview of CryoEM, what's involved from starting with a sample to getting an image on a microscope to doing some image processing to get a structure. So you start with your sample like a purified virus or nanoparticle in a tube. So you have these particles tumbling in solution. And to do CryoEM, you need to freeze it in order to image it. So here, we'll start the movie. So you see the molecules tumbling in solution. You have a 3 millimeter EM grid. You put a tiny droplet of the sample on. We use a robotic machine called a VitrBot to go from the solution to a thin frozen layer of your particles. Then you put that frozen layer in the electron microscope and you shine an electron beam on them and you get projection images. So this is an actual cryo-electron micrograph showing us projections of a symmetrical protein, in this case GRO-EL. So then on the computer what we do is we select out views that look similar, rotationally align them, translationally align them, average them, and get an enhanced view. And then we look through the whole data set and find all the different views. And then once we determine the angles that relate those views, we can generate a 3D structure. So here you see the 3D structure of GRO-EL with features like alpha helices. Now this works very well if your sample is symmetrical and regular. So it works well for icosahedral viruses because they have high symmetry, and unless the particle is broken, every particle will have the same structure. But that's not everything in nature. So there are many things in nature that have a variable or heterogeneous structure. One example is the HIV virion. It's got a lipid envelope with protein spikes and a cone inside. Every single HIV virion is different. They're all unique. So you could not apply the same averaging approach to get a structure. It just wouldn't be a reasonable structure. So another approach using a cryo-electron microscope has been developed called cryo-electron tomography or CryoET. So this is another video by another colleague, Serum Subramaniam, at the NIH. So let's see if I can get this started. So you start with an EM grid. We're going to take a zoom-in view of it. There's this thin carbon layer between the grid bars, and within that there are holes, and they're the size of 1 to 10 microns. And you're aiming to get ice in those holes with your biological sample. So you put a droplet of your sample on an EM grid, and then you use the VitrBot for blotting away most but not all of the water because you want to get a thin layer of ice. So the animation shows you blotting away most of the liquid. Then you plunge it into a cryogen like liquid ethane, so you get very fast freezing, millisecond freezing. Then you transfer that grid into liquid nitrogen, and then you keep it at liquid nitrogen temperature even while it's in the microscope to image it. So this fast freezing at a cryogenic temperature gives you non-crystalline ice, which is important. Now, if you're going to do the CryoET approach, you must add gold particles to your sample, or it helps a lot, because gold is very electron dense and will allow you to image or align your noisy images. So on the microscope, you zoom in on a region where you see HIV particles in a hole. That's your sample. Then you tilt your grid, and you start collecting electron micrographs at various tilt angles on the same region of your grid. Now, these are high-energy electrons, so you have to use a very low dose of electrons so you don't melt your sample while you're doing this. But then you can compute a 3D tomogram. Here's one of HIV. And you can see the pink spikes on the surface of the virus. So then you can computationally extract all of those spikes and average them further, if you know they're the same protein, and generate an enhanced 3D structure. And if you have any atomic information, you can dock it in. And this is a way for looking at the structure, 3D structure of large macromolecular assemblies that you probably couldn't crystallize or get a structure by any other technique. So the field of cryo-EM has undergone a huge revolution in the last couple of years. It's a very hot topic, if you will, right now. And that's because of a technological advance called the direct electron detector. So when I started in cryo-EM, I won't tell you what year, but when I started, most people were using film. They would put film in the electron microscope, and after a day of collecting images on film, they would go in the darkroom for an hour, and they would come out with their negatives of their images they collected. And then maybe the next morning when they were dry, they would see what they had collected. Well, when I started as a faculty member at UCLA, it was early days of the CCD camera for the electron microscope. So I decided to go with a new technology then, and that was get a CCD detector on our electron microscope. And that was a big advance because you'd shine the electron beam on your sample, and within a few seconds, you'd see your digital image on your computer screen. So you'd know right away if you were getting good data. The downside of the CCD detector is it's taking those electrons, it's converting them to light, and then it's converting the light to charge, and then it's counting them in the charge-coupled device, or CCD. So that's a very lossy process. So yeah, we would get a fast image, but it was lossy and noisy. Direct electron detectors, which were commercially available a few years ago, they directly detect the electrons. And you might say, well, what's the big deal? These are 200 kilovolt, 300 kilovolt electrons. They go through a lot of electronic things unless they're very highly hardened to be able to collect such high energy electrons. So it's a big advance to be able to collect the electrons directly, and they're super fast. So instead of collecting a 1-second exposure, we can collect tens or hundreds of sub-second frames very quickly. And that's important for cryo-EM because if you see in the side view of the grid in A, the blue is the frozen water layer and a few virions. Once you shine the electron beam on it, you're melting the ice a little bit and you're breaking chemical bonds. So everything on your grid, even though it's frozen, is starting to move just a little bit. And if you're trying to get a detailed atomic resolution structure, movement is a bad thing. So if you collect a 1-second exposure, you're going to have blur in your image. But if you collect multiple sub-second frames, then you can align them and de-blur them and all of a sudden get a much sharper image like in B that captures the details much more faithfully. And this has allowed the field of cryo-EM to get to atomic resolution, which is important for biologists because we want to know where all the atoms are in 3D space. So when I started in cryo-EM, some people, I won't say who, referred to cryo-EM as blobology because we weren't seeing atoms, we were seeing shapes of proteins. So if you worked on big things like viruses, you could see how proteins interact. But the real goal of biology and structural biology is to see where the atoms are so you can know how proteins interact at a molecular level and how drugs might interact. And so now that has come to fruition in our field of cryo-EM with the direct electron detectors. This is a structure of an enzyme beta-galactosidase at 2.2 angstrom resolution, which is just as good or if not better than a lot of X-ray crystal structures, where you could actually see the density. So the mesh is the cryo-EM density of different amino acid side chains, and then you can place the atoms in there. So this was a true advance in our field. But cryo-EM, as wonderful as it is, it's very computationally intensive, which is why we need the HPC group here. And it's computationally intensive for 3 reasons. First is our images are always noisy. Even with the best detectors in the world, they're going to be noisy because we're shooting these high-energy electrons at a fragile biological sample. It's actually destroying the bonds while we're imaging it. So the best we can do is collect noisy images. So to get a good structure, we have to average thousands, even hundreds of thousands of images sometimes. The second reason it's computationally intensive is that now we all want to collect movie frames because we get better, sharper images. So our last data set we collected at a National Cryo-EM Consortium was 2 data sets. 2 data sets were 12 terabytes of raw data. So yeah, one data set could easily be, 6 terabytes or more. And we only got 4 days on the microscope. We could have, gotten 60 terabytes if we wanted. So, and the third reason it's really computationally intensive is that once you get your structure, you often want to do molecular dynamics simulation, and that is use all the atoms and really do a simulation over time and see how well the atoms fit and agree with your cryo-EM density. So you can use your density to guide the simulation to match what we're observing in the microscope. And that also takes a significant amount of computer resources because the systems we're looking at are on the order of 150 million Daltons. So 10 million atoms and higher. That's without solvent. So, big systems. So now for a few short research vignettes, just to give you an idea of the kind of nanoparticles and viruses we can look at. This is a study we did with Dmitry Shekhamatov, a virologist at Emory University, looking at adenovirus and its interaction with the host factor, which is blood coagulation factor 10. And I'm showing you this paper, even though it's a few years old, because we got it into Science. So adenovirus is a common virus. You've all been exposed to it for sure. It mainly causes respiratory infections. But it's being used in the gene therapy world, and it's one of their common vectors because you can make a lot of it. You can put a large genome into it, and it has useful properties for gene therapy. But when you try to inject this virus into the bloodstream, our bodies are amazing. Our immune system is amazing. Our bodies have ways of very efficiently directing all of the virus, all of the adenovirus to the liver and clearing it from the body, which is fine if you want to do gene therapy of a liver disease, but not fine if you want to treat some other kind of disease in another part of the body. So with Dmitry, we're trying to figure out at the molecular level what is going on to target the virus to the liver so that we could design a new vector that doesn't have those properties or could evade that liver targeting. So we worked with Dmitry on a structure of the virus by CryoEM. That's shown in blue, yellow, and green, different protein components of the virus. And the red is factor 10 bound to the virus. You could see where it's bound immediately in the 3D structure. But then Dmitry asked us, but which residues, which atoms and which residues of the virus are interacting with this factor 10? We need to know precisely. So we did a CryoEM guided molecular dynamics simulation. The red is a model we built for the full factor 10, which is a flexible molecule, docked it into our density, and we had an atomic structure for the virus, so we docked that in light blue. Green are calcium ions that are involved in binding, and we did many simulations. This is just one showing you the final good result. We found there were two residues of the virus that really looked like they were interacting and important for interaction with the factor. And our collaborator believed us enough that he went to the trouble of making new viruses with 3 different residues mutated. Originally we said 3, we narrowed it down to 2, and one of those residues was absolutely critical for the interaction. So you could ablate it and you could disrupt this interaction. So that was one step closer to getting a virus that it can evade liver targeting. Of course, the body's more complicated than that and has at least 2 other pathways for getting adenovirus to the liver, but this is step 1 of getting a detargeted adenovirus vector. In a second project, this is a collaboration with Nicole Steinmetz here in biomedical engineering from nanoscale just a few months ago, and this is work of a graduate student in my lab, Neetu Gulati. So the Steinmetz lab is developing plant viruses like tobacco mosaic virus to be drug delivery and imaging vectors. And her thinking is, yeah, you could use human virus, but you have a risk of disease, so why not use a plant virus? And plant viruses are very highly evolved, stable, complex megadalton structures that would be hard for engineers to design from first principles. So why not take advantage of this assembly and add in chemistries where you can add drugs or modify the virus to do the tasks that you want. And they self-assemble. If you have the genome for the virus, they self-assemble into these large structures. So that's not something really I don't think bioengineers can do. Design a DNA that will assemble into a virus, unless you copy nature. I don't think we can do that from first. Maybe Mike Lewicki could do that, I don't know. But that's a very tough problem. So Nicole starts with viruses like tobacco mosaic virus and can introduce mutations in so that she can add short little polymer linkers, as shown in the middle here, short polymer linkers to the outside of TMV, and then she can add short polymer linkers to a protein she wants to attach on the surface. The red is serum albumin, and then she can chemically link them together so you end up on the far right with a modified form of tobacco mosaic virus. So why would you want to modify it? Well, in this case, we're trying to shield the TMV from the immune system of the body. It's just like adeno. You put some foreign material into the body that you want to use for imaging, the body's going to very rapidly have an immune response and clear it from the body. So if you can shield your particle, make it camouflage somehow, make it look like self, like it belongs there, it will last in the body longer. So Nicole's idea was to add the most abundant plasma protein, which is serum albumin, all over the surface of TMV. So it would look like it belongs in the blood. And of course, there are a million ways you could design this. You could have short linkers, you could have long linkers. So she thought it would be useful to do cryo-EM imaging to actually look at the structure of these engineered particles. So Nitu collected a cryomicrograph. The very dark spots are the nanogold, which I told you about in one of the early movies, so we can align noisy images. And you can see the red arrows mark little black spots which line the surface of these particles. So that's the serum albumin that we're seeing. And there happened to be one TMV marked in white that doesn't have it, and it's got a very smooth edge. So we can definitely see the protein coding on these viruses, but it's 2D. So if we collect a tilt series of maybe 100 different tilt images of that same field, or a new field, we start with a new field, being very careful not to melt the sample before we get to the final image, we can get different views of this specimen. And then we can use those very dense gold particles to align all that information and generate a tomogram, much like a computed tomogram or CT scan of the body. So then we have an actual 3D density map of these TMV particles with the serum albumin coating and the very black spots or the nanogold that we had to have in the sample. But it's easier to look if we do a 3D rendering. So this is a surface representation of that data. You can see the very bumpy surface because there's serum albumin all over the surface of the TMV. And you might say, well, what's what? We can put a cylinder on representing the sides of TMV, make the extra density red, and we still see sort of variably sized proteins shapes on the surface, but some of them match serum albumin very nicely. As you can see, we docked in the crystal structure, and serum albumin is a dimer, so we can do a little more computer averaging because we like using Rogers HPC Center. We can make a template from the crystal structure of serum albumin, and we can search in the density map and say where is there density that matches the structure, and we can only accept we only accept solutions where it fits as a dimer. And we found several positions, and in each case serum albumin is in a random orientation with respect to TMV. So we can quantitatively measure the coverage, and we can visually see how the serum albumin is appearing, being presented on the surface of TMV. And we think that that will allow us to correlate the functional properties of the many different particles Nicole is producing and give her insight into designing the perfect immune-shielded nanoparticle of the future. And the last little vignette is a project with John Pekorski in the Department of Macromolecular Science and Engineering here, and we were fortunate to get on the cover of JACS. And this is work in my lab by Sudhir Malugu, who's our CryoEM manager in the Cleveland Center for Membrane and Structural Biology. So John's idea is similar, but he started with a bacterial virus, in this case called Q beta. It's a spherical virus, and he said, let's try to put different polymers on. And this idea has been around in the pharmaceutical field for a while. Why not put polyethylene glycol polymer on different things to help shield from the immune response? The trouble is, people are starting to get antibodies against polyethylene glycol because it's in all of our cosmetics and all of our products, pretty much. So we need new polymers that are biocompatible and water soluble, and that's what John's involved in designing. So he's tested a few different polymers, and the one at the bottom is abbreviated PNB, or polynorberine. And he did tests of this virus coated with different polymers. He did tests with mouse sera, and he said, which of these polymers provides the best immune shielding? And that was the bottom one, polynorberine. So he came to us and he said, could you do a cryostructure of this virus polymer conjugate? And so Sudhir took him up on it. And viruses are symmetrical, most are symmetrical. So we could get a very nice high-resolution structure of just Q beta alone, but that's not that exciting because we already have an atomic structure of this virus. But on the right, you see we were able to dock in the crystal structure very nicely into the virus. But the part we wanted was the polymer. Now, this polymer is going to be highly flexible, so we're not trying to get an atomic resolution structure because, it would always be changing anyway. So what we want is a general impression of how this polymer can coat the virus, and that's what you can see in the blue and red image. And here it is in 3D. Next time, we're going to do it with a HoloLens. But in 3D, you can see how this polymer coats the virus, and it really only allows small openings that really aren't that big they would be too small for an antibody to squeeze in there and sense the surface of the virus. And so the property of this particular polymer, it behaves more like protein, and it forms kind of a shell around the virus, and it's protecting it very well. So again, we hope that CryoEM can help in visualizing nanoparticles and in designing the next generation, because it gives you a way of looking at the particle as it exists in solution, albeit frozen solution, as long as we have the computers to do the processing. So in conclusion, I hope I've shown you that CryoEM and CryoET are powerful but computationally intensive ways to image things like nanoparticles and biological assemblies. And you can determine a 3D structure one of two ways. You can average images of particles that have the exact same structure, or you can do tilting and get a 3D structure that way. In the 3 projects I showed you, in the adenovirus host factor project, we used molecular dynamics to help understand the interaction, and we were able to confirm the critical residues with mutagenesis. And in the last 2 engineered nanoparticle projects, we were able to visualize the stealth coating and see how in 3 dimensions it's coating the particles. So I'd like to acknowledge the members of my lab, and 2 of my students are here, Linda Thomas and Corey Emerson, and they both have posters if you have a chance to go look at them, both involving molecular dynamics. And I'd also like to thank my wonderful collaborators and you for your attention. Thanks.
So is there any question?
Thank you so much. So how long is this development from the computational world to actual being implemented into a drug, a new drug, for example?
Well, drug companies are taking this approach very seriously. They're laying out $5 million to buy cryo-electron microscopes. So we need more grad students because they're going to be hiring cryo-EM people. Their idea is they could take a membrane protein like an ion channel that's a drug target, freeze it, and get a structure. And if all goes well, people are saying you could get a structure in a few days. But usually it takes longer because you've got to work out sample conditions to get a nice, beautiful image. Then you need thousands of images. Then you need the right software, usually high memory, lots of processors, GPUs are nice. Do some number crunching, then you might find, we need to double our data set. And then, so it's a lot of work to get to atomic resolution. And you need a sample that's very rigid. So drug companies are now doing this. Of course, they won't publish their results, probably, but hopefully that will lead to new drugs because they'll be able to get structures of things that they just didn't have structures for. They were just testing drugs empirically.
That sounds all very exciting.
I'm pretty sure.
Oh, that was fascinating. So I had a question about how do you represent uncertainty in the models? When you're doing the reconstruction, you know in some sense the molecular components, right, that would give rise to the image that you viewed. Mm-hmm. But how much of a problem is that you don't really know, say, whether it was component A, B, or C, or like in the final form?
That's an excellent question. I completely skipped that part of the talk, which you would have loved. The revolution in the field is mainly due to the detector, but also there's been a huge revolution in the software. And the software that's enabling people to get to atomic resolution uses maximum likelihood. So you have to use statistics because you have such noisy data to get out the best structures. If you don't do it, you can still get a structure, but yeah, you could easily go off and get a wrong structure. But with maximal likelihood, it very quickly does 2D classification, throws out the data that doesn't agree with the major class, then does 3D classification against a template structure, and then refines it. And it's just amazing.
Okay. Thank you so much again to Dr. Stewart. Yeah.
You're welcome.